a lot of top-level data collected from the public domain. We are focused on commercial development of novel molecules, so we do exclude biosimilars. And of course, we are focused on antibodies, so we exclude protein molecules that do not have a binding site derived from an antibody gene. I mentioned that data is top level, which means that we track key transitions in the clinical study, certainly, but then the transitions also from phase 1 to 2, 2 to 3, and of course on to regulatory review and approvals. But we do not track every clinical study of every molecule. So from personal experience, I can tell you that the collection of even the top-level data is tedious and time-consuming. But it is interesting and informative to see the results of the data analysis. And when we get to some of my slides, I hope you'll agree with me. And with that, I believe Dr. Lim is introducing himself.
Well, thank you very much, Janice. It's a pleasure to meet all of you. And good morning, good afternoon, or good evening, or chào an,午安, 晚安, 大家. So my name is Seah Lim, and I'm a founder and CEO of Medicovestor, which is a biotech company developing novel antibody drug conjugate. My background is that I actually did my MD training in the United Kingdom, and I'm a board certified medical oncologist, practiced medical oncology until about 2 years ago when I decided to hang up my stethoscope and go into the industry full time. Although before that, I've been with the industry on a part-time basis, on a consultant basis. Okay? And I did my PhD in antibody engineering in way back, a long time ago when we were doing site-directed mutagenesis, and manual sequencing and all that. So, as I say, our company is developing novel antibody drug conjugate, and of course, as a small company, even though our company is small, we have big ambition. We want to make sure that we differentiate ourselves from everyone else. Therefore, we are actually focusing first leverage my expertise in antibody engineering, and we focus on the A of ADC rather than the DC of ADC. And I believe that this is really up the alley of Janice alley. Okay, because you're an antibody person. So with that, I'm just going to pass it back to Janice.
Okay, we have a few slides that should be coming up.
Yep.
Because as I like to say, a graph will cover 1,000 words. So much easier for me to show you the graphs. You can move to the first slide that shows the graph. So I did mention that we have a rather large data set. Well, I think it's rather large. It includes about 2,500 antibody therapeutics that have been evaluated in clinical studies, with most of the development occurring between January 2000 to the present. So with the exception of the approved antibodies, which of course we want to incorporate into the data set, we do exclude those that were developed but had their development terminated in the 1980s and the 1990s, primarily due to relevance and also due to difficulty in finding and validating information for them. So here you see 4 different pie charts, and I'll start in the upper left with pie chart A. That illustrates that more than half of the molecules in our data set, so just over 1,300 molecules, are currently in clinical studies. If I direct your attention over to pie chart B, which is in the upper right-hand side, That illustrates that most of the antibodies currently in clinical studies are in early stage studies, with the outer ring illustrating that nearly 75% of those are in either phase 1 or phase 1/2 studies. It's quite common for antibodies for oncology indications to start, so that is their first in human study is a phase 1/2 study. For indications, those are illustrated in pie chart C, which is in the lower left, with the inner ring showing that indeed about 2/3 of the current pipeline are for cancer. Outer ring showing that about half of those for non-cancer indications are specifically for immune-mediated or inflammatory disorders. We were quite intrigued by the results shown in pie chart D. So now we are on the lower right-hand side, in which the inner ring illustrates that more than 1/3 of the current pipeline comes from companies based in Asia, with the outer ring illustrating that China is the primary source of the pipeline in Asia, even exceeding the number of antibody therapeutics coming from US-based companies. Next slide, please. Following up on that last result, this slide illustrates the growth of the commercial pipeline over time, as evidenced by the first-in-human studies initiated in these periods, with the data stratified according to the company headquarters location. In looking at the bars for North America and Asia, you can see there was a sizable increase in the number of new antibody therapeutics entering their first clinical study in the 2015 to 2019 period, relative to the previous one being the 2010 to 2014 period. But if you look at that 2020 to 2023 period, you can see that there was a rather extraordinary increase in activity in Asia that was not matched by activity from companies based in either Europe or North America. This is the main reason that so many of the antibodies in the current pipeline are at early stage, because they just have not had a chance to advance further. Next slide, please. This graph indeed shows that increase over time again for new antibodies entering clinical studies, but it shows data for each year from 2010 through to 2023, with data stratified according to the general molecular category. The yellow part of the bar represents the standard monospecific antibodies. It's easy to see the increase in the total numbers, but it's also easy to see there the increase in the number of functionalized molecules, such as the ADCs, which are shown in red, and the bi- and multi-specific antibodies that are shown in dark green. By the way, you'll notice there is a decrease in 2023 in the total numbers. That is a real decrease. When I first saw that result, I thought, maybe we missed something. So we did a fairly exhaustive search looking for other molecules that may have escaped our notice, but the trend remained the same. So 2023 was apparently not the greatest year for new antibody therapeutics entering clinical studies overall. Next slide, please. Here, I'm narrowing in on those ADCs, which is the topic of the day. Like the last graph, this one illustrates the trend over time in the annual number of antibodies entering clinical studies from 2010 to 2023, but it's only for the ADCs. So as before, you see the trend towards increasing numbers over the period, but in this case, 2023 was a good year for new ADCs entering the clinic with an all-time high of about 60 molecules. And note, some of them were doubly functionalized in being both ADCs as well as bispecifics. Next slide, please. As illustrated in this graph, we also looked at the trends in the types of payloads incorporated into ADCs. From 2010 even through 2020, most of the ADCs incorporated drugs that inhibited tubulin such as the maytansine and monomethyl auristatin-based drugs, or they incorporated drugs that bound DNA, such as cliciamycin and PBD-based drugs. Since 2021, though, the number that incorporate a topoisomerase 1 inhibitor have substantially increased, such that of those with payloads we know, the ADCs that inhibit topoisomerase 1 comprise the majority. Caveat here, of course, is though that the details of antibody therapeutics can be pretty sketchy early on, and we don't as yet know the payloads for quite a few of the ADCs that entered clinical studies recently. Next slide, please. This pie and bar chart illustrates the frequency of the ADC targets of the current pipeline of ADCs. Pie chart on your left shows the percentages, while the bar chart there on your right show the actual number of ADCs. The most popular target is HER2 at about 10% of the total, followed by TROP2 and Claudin 18.2. B7H3 is the target for about 5% of the total. As shown in the bar charts, at least 28 antigens are targeted by at least 2 ADCs, but please note that, like the payload, the targets for some of the ADCs are not as yet disclosed. Nevertheless, it's interesting to note that nearly 40 ADCs in clinical studies, or about 20% of the total, target an antigen that's unique. That is to say, it's unique, and different compared to any other ADCs that are currently in the clinic. Next slide, please. So I'll conclude with just a couple of slides that show the clinical phase transition and success rates. There's a global success rate for antibody therapeutics. Here you'll see 5 sets of bars with 4 bars each. I'll focus, though, on just the first 2 bars in each set, and those represent the phase transition and overall success rates for standard antibodies, shown in the red, and ADCs, which is shown in the orange. As you can see, the most striking difference is in the phase 1 to 2 transition rate, which is 75% for standard antibodies and 48% for ADCs. So this single transition contributes the most to the difference in the overall success rates, which you can see from the set of bars on your far right. That the standard canonical antibodies have a success rate of 29%, whereas it's 21% for the ADCs, and that is primarily because of that low phase 1-2 rate, because the rates of the standard antibodies and the ADCs are actually pretty similar for phase 2 to 3 to regulatory review, and then review to approval. If you're curious about how this calculation was done or more about success rates, I'd direct you to the Antibodies to Watch in 2024 paper, which was published in MABS in the early part of this year. Next slide, please. This graph, the last one, shows there is a substantial difference in the success rates for ADCs depending on the payload. Pretty dramatic difference, if you ask me. The topoisomerase 1 based ADCs are much more successful so far. So bear in mind that caveat. I do have to point out these rates are, of course, quite preliminary. As I showed earlier, the ADCs with this payload have entered clinical studies in large numbers only recently, so the rates may change as more fates for these molecules become clear. With that, food for your thoughts, I'll conclude my section on trends in the development of antibody therapeutics with a huge and massive thank you to Dr. Silvia Criscioli for analyses and preparation of the graphs. And I now turn the show over to Dr. Lim.
Yeah, let's move forward. Oh, I think Dr. Lim, you're on mute.
Okay. Well, thank you very much, Janice, for the very comprehensive overview. So what I'm going to do is to share with everybody what we are doing in Medicovestor. Okay. Now, to give you a background of where we're coming from, perhaps I'll give you a background of where my training in started. Okay. I was actually trained in Herman Walkman's lab in Cambridge, okay, in the early days of CampPath. So as you can imagine, obviously we were very focused on enhancing the intrinsic antibody property, making use of naked antibody instead of conjugate. Because one of the antibody that I was working on at that point was actually CD33. And at that time, we were trying to make use of, trying to generate CD33 with a strong intrinsic antibody anti-tumor property, rather than at that point, Mylotarg was just being developed. And so that, we are trying to compete with Mylotarg. So through that's the reason why we are biasing the development of our ADC towards the A of ADC rather than DC of ADC. Because I always believe that the A of ADC has been grossly undervalued, has been grossly, has not been fully tapped into the ability. And everybody is just using the antibody, the moiety, as a carrier for the payload primarily. So this is especially more important when you start thinking about now we are going on to primarily cleavable linker, the use of cleavable linker in ADC construct. And if you think about cleavable linker, although initially everybody thinks that well, it is important to actually facilitate internalization of the ADC in order that the payload could be cleaved. But now we have a lot of in vitro data showing that in fact enzymes such as cathepsin B produced by tumor cells, although it's primarily normally within cells, but then now it's been found that it is actually secreted too. So as one could imagine, let's say if you have an ADC that has a cleavable linker that is susceptible to being cleaved by cathepsin B, and that cathepsin B in fact cleave the payload extracellularly, then you're left with a naked antibody that will just be sitting there and that potentially has strong, could be harnessed, the anti-tumor activities could be harnessed to facilitate anti-tumor activities on its own. So this is where we're coming from. Now, speaking as a medical oncologist who practiced medical oncology until 2 years ago, and that all of us believe that ADC, in fact, is the way forward in oncology, because we think that within the next 10 years, no cancer patient will receive chemotherapy as they are being given now. All chemotherapy will be given in the form of ADC because ADC provides higher exposure and also better safety profile. Now, unfortunately, currently we've identified certain gaps in the current ADC because the current ADC relies heavily on the payload or conjugated to the ADC to kill tumor cells. So, as I mentioned earlier, the activity, the potential anti-tumor activity of the backbone antibody has largely been neglected. Now, the other difficulties with transitioning all chemotherapy to ADC is that, like any form of antibody therapy at all, currently ADC is only suitable primarily for tumor cells expressing high level of tumor antigen. Okay? Now, when I make that statement, probably a lot of you will start saying that, what about HER2? Okay, and HER2 has been successful for HER2 low and HER2 negative if breast cancer. However, one have to remember that HER2 or HER2 is kind of a strange, or is a ubiquitously expressed antigen. When we say HER2 negative, essentially what we mean is that when you do immunohistochemistry, the positivity is 1+ or 2+, but without without genetic amplification. Yeah? So there's still a lot of molecules expressed. As recently somebody pointed out to me that in fact when some up to 20,000 molecules of HER2 is actually expressed on many normal tissues. And HER2 negative breast cancer actually express that level or maybe even higher number of molecules of HER2. Okay. Yeah. So as a result, we are actually narrowing the number of patients who could be treated with ADC or even naked antibody, because if that tumor do not express high level of antigen, essentially they're not really suitable for targeting. So in terms of business-wise, you're actually also narrowing the serviceable addressable market. For example, like if you take an example of folate receptor alpha for ovarian cancer targeting, Elahere. Okay. It's actually only capturing around 40% of the patients, because in order to be eligible for Elahere treatment, one actually have to, the tumor cells have to express 75% or more of the tumor antigen. So that's the other gap. Another gap is that as a practicing oncologist and also as a trainee, as a trainer for trainee oncologists, we always teach them that when you give chemotherapy, you always try to give combination chemotherapy, because monochemotherapy invariably leads to development of resistance. Okay? Yet at the moment, most of the or all of the ADC in the clinic or in clinical trial are actually just carrying one payload rather than dual payload. Okay? That's the reason why it's not surprising that response tend not to be as long, the duration of response. So that's the other gap. Now, finally, even though we are now moving towards using humanized antibody, less so with chimeric antibody, okay, the backbone antibody still carries around 6 to 10% of mouse of murine amino acid sequence. So patients still develop anti-drug antibody towards the ADC, although not all anti-drug antibody is actually directed at the ADC, at the backbone antibody, but the majority are actually directed at the backbone antibody. Okay. So what we set up to do is, in fact, to try to overcome all these gaps as much as we can through a novel design of our backbone antibody. Next. So as I mentioned earlier, in order that ADC or any form of antibody-based therapy is more effective, one has to increase the number of molecules of antibody or ADC attached to the tumor cells or to the target cells. Okay. Now, this is largely determined by the number of antigen expressed by the tumor cells. Although one could manipulate the expression of the antigen through some pharmacologic agents, but then doing that also increase the risk of inducing or upregulating the expression of that same antigen with amongst the normal tissue. Okay? So our argument is that, well, we'll try to avoid increasing in the antigen copy number. But what about if for every fixed number of antigen expressed on the tumor cells, we can increase the total number of antibody or ADC associated or attached to the tumor cells? This is a concept that we call functionally increase the antigen copy number. So in fact, we don't really physically increase the copy number. We try to increase functionally increase the antigen copy number by making sure that more molecules of antibody are attached to the tumor cells for a fixed number of antigen. And as a result, we have shown that if you do that, you can actually increase the intrinsic anti-tumor property of the backbone antibody, so that even without the payload, the antibody is able to kill the tumor cells a lot more effective by ADCC, ADCP, or CDC. Now, obviously, when you do that, that you also increase the number of payload delivered to each of the tumor cells for a fixed number of antigen. And so invariably, you also increase the serviceable addressable market. And depending on how you design your ADC or your backbone antibody, it will also give the opportunity, in fact, to actually for you to design combination chemotherapy ADC or ADC with dual payload. And then, of course, if you can use human antibody instead of humanized antibody, then the likelihood for the development of chemoresistance due to the development of anti-drug antibody will be reduced. Okay. Next. So this is our innovation. This is what we do. Okay, we have come up with two platforms. One is aditope platform, as the name suggests, aditopes as in epitope. Okay, and then Antibody platform. Aditope platform is a platform that consists of a pair of antibody. They're directed at the same antigen, but two non-overlapping epitope. So now for every antigen expressed, you have two antibodies, and that essentially equate to functionally doubling the copy number of the antigen. Okay? And that because there are two distinct antibody, it also gives the opportunity, in fact, for using dual payload that are synergistic to each other. And that the good thing with this is that, in fact, you can actually design the ADC in a as long as the affinity for the antigen is comparable between the two antibody, you can actually do a combo or ADC in a ratio of 1:1. So that's fairly straightforward, especially in terms of CMC and manufacturing or process. The other platform that we have is what we call Antibody platform. This is actually a dimeric IgG in which we cross-link the two antibody together. And so now you have increased number of payload delivered to the antigen, and then there will also be more molecules of antibody attached to the tumor cells. Okay. Now we have already shown on with some results that the naked Antibody antibody kills tumor cells with by ADCC and CDC a lot more effective than the monomeric IgG. So with that said, I'm open to any question or further discussion.
Actually, I have a quick question myself. In the ADO-binds version, where you have the dimer there, it's a rather large molecule. There are plenty of versions that are larger than a standard canonical antibody, but that is a full dimer. Are you concerned about the size of that molecule, relative to internalization?
Well, I think that the answer is we have actually in vitro data of internalization showing that the internalization is not impaired. Okay, but having said that, I've always also like to throw out this provocative statement that it very much depends on whether you think internalization is really that important now, with more data coming out saying that perhaps as long as they are in the vicinity of the tumor cells, and then if you have a cleavable linker, then a lot of the payload is going to be cleaved extracellularly rather than intracellularly.
And what about PK, the pharmacokinetics of such a molecule?
We believe that it actually, if anything, it will give a much better pharmacokinetic profile. Because when you think about it, this is a bigger molecule, although not as big as IgM. Okay, and remember, IgM, because of its size, and it stays within the intravascular space, and so it has a half-life of just around 1 week or so. Whereas in contrast, IgG, monomeric IgG, goes everywhere, and it has a half-life of about 3 to 4 weeks. Okay? So this one, we suspect that the half-life is going to be perhaps about the same as IgM, or maybe slightly longer, but definitely shorter than monomeric IgG, because the volume of distribution is going to be a lot smaller, and that this is actually something that we like in oncology, because we like to give chemotherapy to patients and expose the tumor cells to the chemotherapy, and then very quickly remove whatever is not used up. Therefore, to actually avoid the side effects. Like somebody has mentioned to me that the problem with monomeric ADC is that the volume of distribution is so big that, and in fact, especially let's say if the cleavage of the linker is mediated through changes in pH. There will be a lot of free payload being released outside the vicinity of the tumor cells. And then, especially if they are in the extracellular space, they will slowly diffuse back into the intracellular space as time goes by, therefore causing in prolonged side effects. And yet, probably not at a concentration that is enough to facilitate tumor cell killing. That's why we're very excited about Jade, and then we're waiting to start our BKAA study to confirm that. Okay.
And about the ADC platform, can you further articulate various advantages related to it, in addition to, as you were mentioning, about the functionally increasing the antigen copy number? And while you're on the topic, can you also let us know any potential obstacles to the use of the ADATOPE?
Yeah, I believe that ADATOPE platform is not going to be that widely applicable, because as you can imagine, okay, the antibody has to be directed at two non-overlapping epitope. Okay, and again, it's a reflection of my age. I'm used to immunizing mice. And then especially if you use an immunogen to immunize mice, you invariably end up getting antibody just directed at the dominant epitope. I remember that when I was working with CD33, I was working hard I generated a big panel of antibody, CD33 antibody, to try to identify a synergistic epitope, a synergistic pair of antibody. And the panel of antibody that I generated was through immunization. And we did not manage to find a single one. Okay? Now, we have been very fortunate. At the moment, we have at least one asset that actually has a pair of antibody that are suitable. And that's actually through the use of not immunization, but through the use of phage display library. But of course, you know that the problem with phage display library is that generally the affinity tend not to be that satisfactory. So you might have to go through affinity maturation. And even with the phage display library, we managed to isolate 35 distinct clones directed at one antigen. And then after screening, we actually only have one antibody that was directed at a non-overlapping epitope. Okay, so that's one difficulty. The other potential obstacle, obviously, is toxicity. But then you could make the argument that, well, if you use synergistic pair with payload that are synergistic, you may not need to use that high a dose of your ADC. So, and it's obviously something that one has to be aware of. Perhaps one way to go around it would be that one could envisage, it depends on whether you want to enhance the intrinsic factor, the intrinsic antibody function or not. If you do not want to, you could easily say, use your clone your V gene onto IgG4 instead, Okay, perhaps if you use IgG4, you probably have to use the mutation to prevent arm switch or reduce arm switch age. And then that may reduce the non-specific FC binding because I think that that is, as an oncologist, that is probably the most the thing that we worry most, because we worry about myelotoxicity. Because once you've got myelotoxicity, that actually, although now we've got hemopoietic growth factor support, but then that actually may delay the next cycle of your administration of ADC.
And we could have you join us again and let us know if there are any general questions or questions from the general audience.
Yeah, so if anyone that's listening, please feel free to shoot your questions right now, and we'll have the last couple minutes for the speaker to address them.
Yeah, what I can say for my part, while people are thinking of questions, is that we do collect an awful lot of data, and we do make at least some of it available directly on the Antibody Society's website. So if you are interested in exploring what actually is already approved and what's in regulatory review, we have a searchable table right on the Antibody Society website. If you're curious, well, which ones are the tubulin inhibitors and which ones are the cliciamycin based ADCs or the topoisomerase based ADCs, you'll be able to tell that right from the table. And indeed, we also have the late stage pipeline on the website too, and a similar table that is searchable. So you'll be able to find that information, at least about the late stage pipeline, what's in regulatory review and what's already approved. The rest of the pipeline we don't have online yet. You'll have to wait a little bit. We're working on it though.
I think that I hope that today I could impress on everybody that a component or a moiety within the ADC that's been that's been largely neglected. Okay, because really, when you speak to everyone else in the ADC space about ADC, everybody is just talking about payload and linker. Okay? And that, yeah, people are saying that, well, we've got linkers that are more specific because it's pH sensitive. But then when you think about it, as I alluded to earlier, okay, when you have a monomeric IgG that goes into the intracellular space where the pH is different, okay, and so right away you are actually not really, if anything, you are making it less specific than more specific. Okay? And that that's why, I mean, it fits in well. Our what we're doing, it fits in, fits into my expertise well what we're doing in our company, because I love antibody, and that I think that biologics have a big role. And I feel that it's not just the anti-tumor activities of the backbone antibody through ADCC, ADCP, and CDC, but you must also remember that when there's complement activation, there will also be associated cytokine that again provides further anti-tumor activity. That's why we are very excited that we've been able to go into a niche within ADC that no one else is doing. And of course, it's up to us to convince everybody that this is the right way forward. I mean, there's not any doubt at all that one also, there's a role in optimizing the payload and the linker. But then we feel that, before we do that, why don't we just optimize what is obvious first?
Okay, I just saw like two questions popped up, and I want to share that with the speakers. So one is the ADCs has received a lot of attention lately in the scientific investment community, and that's definitely for sure. We also notice a lot of the transactions and deals that's happening in this space. And have you both noticed a spike in interest or request for due diligence from VCs or investors?
Well, I wouldn't really be able to divulge that. But yeah, absolutely. It's a hot area. I mean, it has been around for quite some time. Immunogen's been working in the ADC space now for, 30 plus years? But there has been tremendous expansion in the types of antibodies, the antigens, as Dr. Lim was referring to, the linkers as well as the payloads, and that has allowed a lot of additional companies and very creative scientists to also engage in this kinds of activities. So there's way more action, there's way more possibilities, so there's way more interest because they are becoming more successful, especially with the topoisomerase 1 inhibitors. You see a much more successful so far. And so with these additional possibilities, you have more interest from investors, more interest from the major pharma firms who don't want to have to invent everything themselves. That's what biotech does very well.
Yeah, I think that I would echo what Janice say. And certainly to further emphasize, in fact, when I spoke with my colleagues in medical oncology, all of us believe that within the next 10 years, all chemotherapy will be given in the form of ADC. Because now we are seeing more successes with ADC. And that definitely there's a lot more interest from the VC if you look at the recent and also from investors and also from big pharma, because if you look at the acquisition and deal, the M&A deal in the last 12 months or so. Okay? And as far as my company is concerned, certainly we are seeing some interest as we speak. Actually, we are undergoing DDs from 2 VCs at the moment. And yeah, I think that I'm glad that there are VCs that actually appreciate or understand the scientific basis of what we're doing, whereas a lot of the people just think that an antibody is an antibody. Okay, so I'm really glad that we have the opportunity to really impress on them, and that I think as we move forward, there is the opportunity to actually, show that what we're doing, in fact, could be applied. Our platform could be applied to any target, could be applied to any antibody at all. I think the difficult thing in terms of the industry is that, in my experience, there are 2 different groups of investors. There are the investors who always say that, well, we want you to go for novel target. There are another group of investors who say, no, we want you to go for validated target. Okay? So you can never satisfy the 2. So I think that it's just like I always joke saying that, everybody eventually, or most people eventually still find a partner. And it's just that, beauty is in the eye of the beholder.
Yeah, there very is. That what you mentioned is very true. There's been arguments for ages over whether, especially in biotech space, whether you should use your technology against a well-validated target because there's lower risk associated with that, but isn't nearly as much differentiation. There's much higher risk associated with an unvalidated target, but potentially a much larger reward also.
Yeah. Well, to follow on with that point, when we spoke with a very the JP Morgan and asset management team, what they said to us, because actually we are playing with two targets in our company. Okay, one is validated target, folate receptor alpha. The other one is a novel target. Okay? And that we were told, don't go for the novel target because your backbone antibody is already novel enough that you don't want to take another extra layer of risks. Yeah. And then I'm quite sure most people would appreciate that, as like most of the small biotech company, if your first product fails, that's the end of the company.
Well, yes. Thank you again for the insights, and this is definitely, the first-hand experience on this end, and also best luck on the due diligence process.
Thank you.
And yep. And also have like one other question before we end this session. And so David asked, what the challenges exist in the manufacturing of ADCs? What are the key considerations in the scalability of the production process for clinical or commercial use? If you guys have any insights on this area.
I'd have to defer to Dr. Lim on this.
I think, in my mind, it depends. Let's say if you're doing monomeric ADC, it should be fairly straightforward. Okay. However, if we're talking about antibody, like our platform that we're doing, it's less straightforward because the dimerization is done, outside the cells, extracellularly. Okay? So you actually have to go through an extra step. But then, having said that, yes, we have found that we have adopted method that could be scalable. So we're very excited about that. Yeah, I mean, it's very easy to say we do this is in our lab, and then, when it comes to the scalability, it cannot be done, then it's still not translational.
Awesome. And yeah, and also if anyone have further questions about this one, we're also having our internal speaker from Asun Bio coming on to the event in November to address questions around the manufacturing capability. So, stay tuned for that as well.
Yeah.
And then I think we are actually right at the time for our 1 hour mark.
Can I make one last comment, Okay. Yeah, I'll be more than happy if anybody have any question and to contact me directly. You can make my email address available to anyone. Okay.
Sounds good. I will insert your contact information in the follow-up, thank you email, and for all the people that signed up for this event.
And of course, people are welcome to contact me too. And for my side, I'm perfectly fine if somebody wants the slides. So if you want to include the PDF of the slides, that's fine with me. Dr. Lim, I leave that, your part of it up to you.
Yeah. Okay,
Okay.
same here.
Okay, sounds good. Then I will, update those information and will insert your contact information in the follow-up email. So make sure every audience has it. And we will also, we can talk more offline to see how we can insert your contact information in our other platforms as well.
Well,
Okay,
thank you very
good.
much. Appreciate the
Thank you very much,
opportunity to be here.
everybody. Have a good day.
Thank you.
Yeah, have a good one.
Bye.
Have a good day and have a good night.
Bye.
All right,
Bye.
thank you. Bye.